presenter is Alan Moses from my department, Cell and Systems Biology. He was recently tenured, and he's at the forefront of understanding regulatory motifs by developing sophisticated statistical models. So he will be telling us about short linear motifs, a long-range drift from bioinformatics to systems biology.
Okay, well, thanks, Nick, and to TorBUG for the invitation. It's great to present here, and also to Shobit because he gave a great introduction to what I'm going to tell you about. So I'm going to try to tell you a lot of stuff in a short time. I hope that it will be mind-expanding, at least because of the pace. I want to give you a short introduction to short linear motifs very quickly, and I'm going to tell you about really mostly experiments that we've been doing in our lab to test our bioinformatics predictions. And then I want to get into more bioinformatics, where we started getting into evolution, and then how we started trying to test those predictions. And finally, if there's time, I want to tell you about how we can start to use short linear motifs to tell us about the evolution of novel signaling interactions, which I think is a very exciting prospect. So we already heard from Shobit that short peptides are really the key points in signaling pathways that connect the signaling pathways together. Here are some examples of these proline-rich SH3 binding sites that Shobit was talking about. This is this phage display. This is a representation of this phage display data from Gary and Dev. These are other examples of different kinds of short linear motifs that mediate different kinds of interactions in the signaling network. And these motifs fall into families, so we can represent them this way. But there are other short peptides that also are important for interactions in these pathways that actually don't seem to fall into sequence families, and they might be unique in the proteome, and less is understood about these kind of motifs. And I'm going to tell you, I'm not going to go over this very much at all today, except to say that a few years ago, Alex Neumbaugh, when he was a grad student in my lab, developed a single feature, namely evolutionary conservation. He showed that single feature could identify 30% of the previously known 350 motifs that we found in the literature. Okay, so that's just a single evolutionary conservation feature. So it seemed to us that evolutionary conservation could be an incredibly powerful predictor of these short motifs in signaling proteins. And so we wanted to actually test whether this was really true. And so the way we chose to do that is using a model signaling pathway, and this is the HOG pathway in yeast. You can see the clever titles, but the point is that the HOG pathway is important for the cell stress response in response to salt. Okay, and again, it's a well-characterized MAP kinase pathway, has lots of disordered regions, signaling domains, and we just decided to look at the phylogenetic predictions in this pathway. Anyone can do this. We have a website. It doesn't look that nice, but you can just type in your favorite protein's name, and magically we predict in bright red boxes here the short linear motifs in your protein. Okay, and we used our own prediction method. We just took it off of our website, and we predicted 29 of these things in our pathway. Okay, and in this model HOG1 pathway. And so here's a schematic of the pathway. These are all the known components. This is the MAP kinase cascade. I'm indicating these wiggly lines to show you that there's these disordered regions in the pathway. And this is an example of one of these known SH3 interactions that Shobit was just telling you about in this signaling pathway. And these are just examples of our kind of predictions. These are these highly conserved short pieces of the protein that are conserved over long evolutionary distances. Okay? And so in order to test these things, we needed an assay in the lab. So Bob Strom in the lab developed a very straightforward transcriptional reporter assay, where if we treat we hook up a promoter that's responsive to signaling this pathway to GFP, we treat the cells with salt, and we watch what happens to the expression of this reporter. And what you can see here in this flow cytometry data is the reporter expression is moving this way. It's increasing when we treat the cells with salt. If we don't treat the cells with salt, the reporter expression doesn't change. These are just 3 overlays of replicate experiments where we're putting the cells through the flow cytometry. Okay, so we're going to do this kind of experiment over and over again. So I hope everybody is okay so far with the flow cytometer. So what kind of experiment are we going to do? So this is the basic idea. We predicted this bioinformatically based on evolutionary conservation. We go ahead and we just make a small deletion in this motif, which we can do in yeast, and then we just go ahead and redo this experiment and ask, well, now what happened to our reporter expression? And in this case, I'm showing you this is our best case. This is an unknown short motif that we predicted bioinformatically, and deletion of that motif completely abolishes the cell's response. Okay? So this is an example where a short linear motif actually is critical for the signaling through this pathway. Okay? And because we're getting this flow cytometry data, we thought, well, we can actually quantify the cellular response, and it's going to become important for what I'm going to tell you about. And so we can quantify the effect of mutation. So here's a bunch of known motifs that we knocked out responding to salt. Here's all those same things when we don't treat with salt. And then what we decided, with the help of Mitch, a grad student in my lab, we decided to compare these two histograms to calculate both the fraction of cells that turn on, which we define as the cells that move beyond the distribution of cells that we see in the no salt case, as well as, given that these cells do turn on their GFP expression, how much do they turn on? Okay, so both the fraction and the amount that the cells are going to turn on. And then we actually display this data as follows. So we plot the fraction of cells that turn on for a given flow cytometry experiment on this axis, and given that the cells do turn on, how much do they change their expression? Okay, so here's the wild type cells. These are many replicates of the wild type experiment, and here are individual replicates of this mutant that I showed you that nearly abolishes signaling. So this is obviously a very dramatic effect. But of course, I'm plotting it this way because I want to show you that not all mutations have this as dramatic effects. Here's other known motifs. Some of them have big effects, some of them have smaller effects, and even still, some have even smaller effects. And in fact, most of the mutations, most of the short motifs that we deleted fall into this class that have sort of quantitative or moderate effects. And we can zoom in on this sort of cloud of points here and show that actually some of these motifs that are showing relatively small effect, actually they are showing statistically significant effect. So we think that there's a lot of short motifs in these signaling proteins that are actually having statistically small but quantitative effects that are important for signaling. Okay, so just to summarize what we found from this experiment, from this sort of systematic experiment, of the 15 bioinformatics predictions that we tested, 6 of them actually showed effects on signaling function. Okay, so that we think actually of the total 29 predictions, 10 of them are real motifs according to our assay, and so that's really the lower bound on the number of motifs that are out there in this pathway, and we don't think there's anything special about the HOG1 pathway. There's lots of other pathways like this in yeast. For example, we did a similar calculation for this hypoosmotic stress pathway, and in fact it has 36 of these predictions. You can go and look in human, and the Wnt signaling pathway has 28 of these predictions, and we think, so there's lots of these short motifs out there. Most of them, we believe, remain uncharacterized. Okay, so that was great. Our bioinformatics is working, but as we were doing this kind of quantitative experiment, we noticed some very interesting phenotypes, and this started getting us on the road to systemic biology. So what, if we look at this mutation here, and I'm losing, unfortunately, my battery out here, but if we look at this, this mutation here, what you can see is that this mutation is having a very small effect on the change in reporter expression, but it's having a significant effect on the fraction of cells that are responding. And that's highlighted here in the histograms from the flow cytometry data, where you really do see two populations of cells. That some of the cells have turned on their reporter expression, and some of them have completely failed to turn on their reporter expression. I'm having bad luck with these things. Oh, this one's really dim. Some of them have completely failed to turn on their reporter expression, and that's because there's some kind of bimodal phenotype here. And so this is very interesting, and at first we thought, well, maybe this is some kind of artifact. So we did. So Bob did the following sorting experiment, where he sorted out the two populations of cells, those that respond and those that didn't respond. And then he repeated the exact same experiment again, treating them with salt and comparing those that were untreated with salt. And what you can see here, where it says secondary induction, is that we recapitulate these bimodal distributions of cells. So what we're getting is that this isn't some inherent property of this. This is an inherent property of the cell population. It's not like half the cells are dead and half the cells are alive. Each of these individual populations can recapitulate both of these two responding and non-responding populations. Okay, so this is very suggestive that there's some kind of stochasticity noise in the response of this pathway. And just to show you that it's not, we also considered that maybe there was some problem, we were just not waiting long enough, maybe these cells were somehow delayed in their response. So here's just letting the cells go for another hour or another 2 hours, and you see that you still never, these cells that are sort of off, they just don't turn on. So there's something about these cells that they fail to respond to the signaling pathway, and we thought, well, maybe the cells are, the salt is killing them and they're exploding or something like that. So we went ahead and looked under the microscope, and sure enough, the microscopy recapitulates the flow cytometry, that there's some cells that have really bright reporter responses. Those cells, I hope it's clear, are these, the green cells, and then there's some cells that simply have what looks like negligible reporter expression. So it really looks like we are getting these two populations of cells. And again, we were trying to rule out any possible weird effects here. Maybe we were messing up this protein in some way. So Bob made another mutation where he just mutated 2 residues within this short linear motif. And again, we repeated the experiment, and we still see the same 2 cell populations. So it really looks like there's something specific about this motif that's leading to this stochasticity, okay? And now I told you this as a sort of surprise story, but in fact, in this pathway, this model map kinase signaling pathway was already known to have stochastic responses in some cases. So people, systems biology guys, had reported that there was evidence for this kind of stochastic behavior in this pathway, where some cells would respond and some cells didn't. And in fact, in these papers, they went so far as to propose models for how this was actually happening. So let me just walk you through one of these mechanistic models from one of these papers. Okay, and again, this is a stochastic model, so each of the reactions in this model is going to be allowed to go according to some kind of a random process. And so here's the actual pathway, or at least the schematic of the pathway I showed you before, and here's the model of the pathway, okay? And so what you can, I hope is clear here, there's no MAP kinase cascade here. There is some kind of stress and response to the stress. So there is a parameter in the model that controls the strength of the stress, but there's no MAP kinase pathway. In the real pathway, there's all these complicated protein-protein interactions, and in the model, there isn't any of that. There's just a cycle of phosphorylation and dephosphorylation of the MAP kinase in the pathway. And then most of the model is actually dedicated to the transcriptional steps, the interaction of the transcription factors with the promoters, and the interaction of the polymerase with the initiation complex and the formation of some kind of active gene. And that is all modeling these parts of the pathway that I didn't talk to you too much about, but the reason they focused on that part of the pathway is because it's thought that they argued that the stochasticity and the noise in this pathway is really coming from these slow transcriptional steps at the bottom of the pathway. Okay, and I should say the mutations that we found that were causing these bimodal or stochastic effects were actually in proteins that were in this stage of the pathway. So this was all very consistent in some ways with the model that they proposed, although of course they didn't point to any connection with short motifs in the signaling pathway as underlying the stochasticity. Okay, and just to try to convince you, so I managed to persuade Ian, a graduate student in my lab, to actually implement this model. And so here's the output of one of his simulations, where we actually have, you can see that what the stochastic model produces. It's a number of it gives you an exact number of protein, reporter protein molecules with each simulation. And then we do the simulation many, many times, imagining that each simulation corresponds to one cell going through the flow cytometer. And maybe it's not very convincing, but this kind of output looks a lot like the data that we're getting out of our flow cytometry experiment. So the great thing about this is that we can then take the output of this flow cytometry experiment and run it through exactly the same quantification scheme that we used on our real data. So maybe I said that wrong. We can take the output of our simulations and run it through the exact same data analysis pipeline that we did for our flow cytometry experiment. So we think that we should actually be able to compare the predictions of the simulation to the output of our experiment. So first, let's see what this simulation says. So one thing we want to do is we want to actually make mutations. But of course, the proteins that we actually mutated are not in the simulation. So how do we model the effect of mutations in the pathway. Well, so what we did is we just took each parameter and we systematically varied the parameter. Okay, and so here's the result. So what Ian plotted here is that as you vary each of these parameters in the model, and I'm just indicating them with those numbers 1, 2, 3, 4, they correspond to different parameters in the model, you could plot out what effect that has on the model prediction. And again, we're doing the exact same experiment in the model. We're comparing salt to no salt and running through the exact same data analysis pipeline, and we're extracting the fraction of cells that actually turn on and the change in the GFP intensity for the cells that turn on. Okay, so it's exactly the same plot, except now from this the output of this stochastic simulation. And what you can see here is that mutations in the parameter that control sort of the pathway, that is how the MAP kinase actually gets turned on, fall sort of along this line, which affects really both of the fraction of cells that turn on and the change in GFP intensity, whereas these traces all correspond to parameters that are a part of this transcriptional process. And again, this is consistent with the idea that that transcriptional process is important for the stochastic effects here, because what you can see is here those changes in those parameters tend to affect the fraction of cells that turn on more than they affect the change in the GFP intensity. Okay? And then you can see there's some other parameters here that I'm not going to talk too much about that have other and sort of more complicated effects. Okay, so now we can go ahead and compare what the model is saying mutations in these parameters could have to our actual data, which I already showed you. So now I'm overlaying the data onto this plot. And what you can see is that for some of these predictions, first of all, you can see that qualitatively the model does predict pretty well the correlation between the fraction of cells that turn on and the actual change in GFP intensity. But the main point that I want to make from this figure here, and it's too high for me to reach, but this mutation that showed this very strong bimodal phenotype in our experiments actually is even more strongly bimodal than the model can predict. So that even though the model does predict these strong stochastic phenotypes, it always predicts that there should be some change in intensity of the GFP expression, even for the cells that turn on. Whereas we have this mutant that shows actually almost a full, essentially a full pathway induction for the cells that do turn on, and cells that don't turn on show almost no expression. Okay, so just to summarize, we think that these we can go through these conserved segments in the disordered regions of signaling proteins. Mutating them affects the signaling pathways in interesting ways. In 1 out of 6 of the new motifs that we identified that could show statistically significant effects in our assay, shows this very strong stochastic or bimodal effect. And it suggests that actually these things are probably not very rare. There's probably lots of mutations out there that are having these stochastic effects on signaling. And I mean, this is, of course, very interesting. So again, this is another one of these systems biology kind of papers where they suggest that the variability or the intrinsic, this kind of stochasticity in gene expression could be a mechanism for incomplete penetrance. And if you actually look at where disease mutations fall in the human genome, many of them actually do fall in disordered regions, and many of these mutations are known to be incompletely penetrant. So we think this is the first sort of connection between the mechanism of stochasticity and disordered regions and motifs in those regions. Okay, so I gave you a short introduction to linear motifs. I showed you systematic predictions of our systematic tests of our bioinformatic predictions in a model pathway and how that led us into these kind of systems biology considerations. So now I do have time to tell you a little bit, I have plenty of time to tell you about some of our, how our bioinformatics got us into thinking about evolution of ancient regulatory networks. And in this case, we're going to think about this paradigm of gene duplication and divergence, but in the context of these short motifs, okay? So I showed you this diagram already. It already looked like a big mess, and that was the diagram for budding yeast. Again, these are the MAP kinase pathways. If we look at the mammals, of course, these are the highly conserved MAP kinase pathways, but I hope what's clear to you is that the mammalian pathways have a lot more things. They've got these it's an even bigger mess than it is in yeast, essentially, is the point here. And then we have a hypothesis that this increase in complexity in the mammalian signaling pathways might be due to changes in the short linear motifs that are important for these wiring of these signaling pathways, okay? And there's a lot of precedent for thinking about evolution of regulatory sequences. So regulatory sequences can influence evolution at many different levels. So for example, transcriptional regulation, where regulatory sequences in the DNA are important for evolutionary diversity. Transcription changes in splicing can be important to affect the proteins that are actually produced in different species. And then finally, we're at the post-translational level, where changes in short linear motifs and post-translational modifications can affect the regulation of proteins in different species, okay? And there indeed have been some examples, although there are very few, of adaptive post-translational regulatory evolution. And this is just an example of one in this paper. Okay? So the paradigm that we're going to think about post-translational regulatory evolution is in this gene duplication and divergence paradigm, where we have an ancestral protein, it duplicates, and then the two extant proteins diverge. And if we imagine that these proteins were transcription factors, we might think that the ancestral protein bound to a certain DNA element it had a certain recognition site. The proteins duplicated and diverged, and now there was a slight change in the specificity of one of the transcription factors, so that it now regulated slightly different target genes. Okay, so now this is sort of what I call the traditional view of how a protein could duplicate and then slightly diverge its function to take on new functions over evolution. Okay? And I want to contrast that with what I call this idea of post-translational regulatory evolution, where an ancestral protein can duplicate, and then in this case there's a short linear motif or maybe a phosphorylation site in that protein, and that protein could duplicate, and then the extant proteins, one of those short linear motifs could diverge, and that could lead to a change in the post-translational regulation of the protein, but leaving no change in the actual DNA binding specificity, for example, if these were transcription factors. Okay, so that's how proteins could keep their biochemical function the same, but they could diverge at the post-translational regulatory level. And of course, you can imagine more complicated scenarios where you actually have multiple motifs in the ancestral proteins, and then after gene duplication, different copies retain different short linear motifs, leading to this evolutionary model of subfunctionalization. So what could we, how could we get at this bioinformatically? Well, what we could do is use our bioinformatics methods to first identify the very strongly short conserved sequences, okay? And then we could look in species where those proteins, where we found the conserved short sequences duplicated, and see what happened to the conserved sequences over evolution. Okay, so here's a specific example of that. I already showed you these two transcription factors. There was a single copy ancestral protein. They duplicated. Okay, we can use our bioinformatics methods on these outgroups where these are species that didn't duplicate the gene, and now we have a short conserved peptide in those single copy proteins, and then we look at what happens after gene duplication, and sure enough, we can find cases where the short regulatory sequences have rapidly diverged along the lineage, leading to one of the paralogs. Okay, and in this case, as I already mentioned, these are transcription factors, and sure enough, these are known regulatory sequences in one of the extant transcription factors in bodies that have been lost. Well, we figured out that these have been lost from the ancestral protein. Okay, so this is a case where you duplicate and then you lose regulation in one of the paralogs. And in fact, there seem to be lots of examples like that. So here's an example of a phosphorylation site for HOG1, which we already talked a lot about, where in one of the paralogs after gene duplication, it's lost from one of the paralogs. And that's indicated by the red boxes in these figures here. And here's another example of another protein that's regulated by the ancestral protein was probably regulated by this kinase CDK1. There was gene duplication, and one of the paralogs lost the phosphorylation site. It's probably not regulated anymore by CDK1. Okay, and I want to now spend the rest of this section telling you about what we think is the most spectacular case of gene duplication and post-translational regulation that we've identified so far using these bioinformatic methods. And that is this case of BUG1 and Matthew. Okay, and something important to say about these things is that these paralogs in Saccharomyces cerevisiae are part of an incredibly ancient signaling network that's part of the spindle checkpoint path. It's part of the spindle checkpoint, okay? And this is something that happens in every cell cycle, and it's critical to the proper segregation of chromosomes, okay? And so what it turns out is that there was a single ancestral protein that had all these functional elements in it, and there was a gene duplication and divergence where one of the paralogs lost two short linear motifs, in this case these chem boxes, and the other paralog lost the kinase domain. Okay, so the ancestral protein is the kinase. Now we have one protein that's a kinase and one protein that is no longer has the kinase, but now has these two regulatory sequences that were found in the ancestral protein. Okay, so and again, we know a lot about this spindle checkpoint. We know a lot about the signaling network. This is what we infer the ancestral structure must have been, and this is what we think the extant structure is. And again, of course, these are greatly simplified regulatory checkpoints. Okay, I'm going to skip this. But I want to show you something really what we thought was really impressive. So here is yeast growing on plates, and then what happens if you treat yeast with a drug that damages its DNA, and the spindle checkpoint becomes really critical for cell survival. What you can see is if you delete either of these proteins from the spindle checkpoint pathway, the cells die. If you delete both of them, you won't be surprised that the cells also die. But the point here is that if you replace one of these proteins with a single copy protein from one of these species that didn't duplicate, in fact, it can rescue the function of both of the extant copies. Okay, so the single protein from a species that has one protein apparently can do both of the jobs of the two extant proteins. Okay, so you might not be so surprised yet, but let me try to show you some more interesting things about these things. So the other thing is you can look, these proteins have diverged quite a bit. You can look at their localization patterns by GFP tagging the proteins. Here's some microscopy that was done by an undergrad research assistant in our lab, Selma, and what she could show is that you see these, for the BUG1 GFP tag protein, you can see these very nice kinetochore dots where the protein is right at the kinetochore and only very early in the yeast budding cycle where there's very small buds. Once the buds get bigger, you really don't see this protein anymore. Okay, if you look at the GFP pattern for MAD3, what you can see is that there's a constitutive nuclear localization, and you can very beautifully see the MAD3 in the nucleus throughout the whole cell cycle. Okay, and now we can go ahead and make a fusion protein of this single copy protein that's from the species that didn't duplicate their BUG1 and MAD3. And what you can see here is that this protein actually shows a combination of both patterns of the single copy protein. So that you can see it in the nucleus throughout the cell cycle, but during the early stages of the cell cycles, when BUG1 is normally found at the kinetochores, this single copy protein is also found at the kinetochores. Okay? So this looks to be a classical case of subfunctionalization. And you might say, well, that's actually the leading paradigm for gene duplication and divergence. So maybe, okay, so you showed some GFPs and stuff, but this is sort of what we already expect. But for those of you who are spindle checkpoint aficionados, you will already have realized that there's something funny going on here. And that is that it's well known that humans also have a MAD3 and a BUG1. But now I'm telling you that I can find yeast species that diverge from Saccharomyces cerevisiae before the duplication of these proteins. So how is it that humans also have a BUG3 and a BUG1 and a MAD3 when this protein supposedly just duplicated in the ancestors of yeast? And it turns out that this same gene duplication and divergence has happened many, many times over evolution, okay? And that, I hope, will be surprising to everybody. At least it was very surprising to us. Unfortunately, this observation was published in 2012. After, of course, we discovered it, but we didn't publish it in time. But the point is that the same the same ancestral protein has duplicated and diverged many times throughout the animals, through other fungi, and even in Arabidopsis. Okay, so this is some kind of pattern that is happening over and over again throughout eukaryotes. The same gene is duplicating and diverging. And it goes even further that in many of these gene duplication cases, you see the same consistent division of function, that the kinase is lost from one copy, and these Ken-box regulatory short linear motifs are lost from the other copy. Okay, and you can see that in the bioinformatics prediction. So when you see something happening over and over and over again in evolution, it's usually thought that lightning doesn't strike twice, and that this must be some kind of adaptive evolution. There must be some kind of benefit to duplicating these genes and actually changing them in precisely this way. Okay, so that is what Alex set out to test. Okay, so how do you actually test that these changes were actually beneficial to the organism? How do we actually test that kind of hypothesis? And the way you can actually do this in biokinesis, you can actually directly compete yeast strains in the lab. Okay, we know that the spindle checkpoint is going to be important, especially when we stress the cells with these drugs. And we can actually ask, how well do the cells grow in the lab when we treat them in various ways? And the way we can follow the growth of the strains, we can do direct competitions by labeling one strain, for example, in this case the wild type with RFP, and another strain with GFP, in this case the mutant. Maybe this kind of assay is very familiar to all of you, but there's a very strong internal control for the growth conditions of media, etc. And what you get out of this is, so you put the cells, you grow a mixed culture of your mutant and wild type cells, and then you grow them and measure the size of the cell populations in the flow cytometer again. And so what you can get out of this is the ratio of green cells to red cells as a function of the generation, and that's very easy to convert back into a selection coefficient. And what I'm showing you here is that we think we can measure very small selection coefficients with this assay. So fitness effect of less than 1%. Okay? So now we can go ahead and ask the question, well, was this gene duplication and divergence actually beneficial? And the amazing result, at least to us, is that we can't find any evidence that this was actually beneficial. So let me just show you what the data are. So what we're showing you here is that our estimate of the fitness or selection coefficient. Okay, 0 is indicated here. This gray curve tells you the resolution of our assay. So what we did is we did 64 comparisons of genetically identical strains against each other. Okay, and this is the distribution of selection coefficients that we actually get. Here are the single deletions of these genes. Okay, so the double deletion is even more dead. These single deletions, one of them is very, very dead. One of them, we can't really even measure its selection coefficient in this assay. The MAD3 deletion alone has a severe reduction in its growth rate and its fitness in this assay. But when we rescue the double deletion with the single protein from one of the other species, the measurements we get fall within the distribution of genetically identical strains competed against each other. And the reason I have both a green and red bar showing you where the result for each of these genes is because we can do each of these experiments and we swap which strain is attached to RFP and which strain is attached to GFP. So that shows you the kind of inter-replicate variation that we're getting in this experiment. Okay? So as far as the resolution of this experiment, we can't find any evidence that having the two extant yeast proteins is actually better for the cells than the single copy protein. And again, this should surprise you. We're taking a protein from another species, and it seems to do just as well as these two duplicated proteins. And again, those two species have diverged by millions and millions of years. Okay, so again, this was very surprising to us, and it made us think that this must be some kind of example of neutral evolution. But again, this is very surprising to us because when we see evolution happening the same way over and over and over again, it's usually thought that that's called convergent evolution, and it's usually thought that that's evidence for natural selection. And in our case, we think that actually it must be some kind of neutral process. So we wanted to explore this even further, and so we did the following experiment. Okay, so what I've been showing you so far is that the extant cerevisiae configuration, we can replace both of these genes with a single copy protein, and we can rescue the function. But the question is, well, evolution doesn't happen in that kind of way. It has to happen in some kind of stepwise process. So maybe there's actually no way to get from there to there, and something much more complicated has happened. Okay? So then we set out to use, again, another technique that was developed in Brendan and Charlie's lab, to use automated genetics to actually make lots of evolutionary intermediates. So what we can do, for example, is we can say, well, what happened? The ancestral single copy gene duplicated. Okay, so then we can make a strain that has 2 copies of that single copy protein, and then we can ask, those 2 copies then could diverge. We don't know how they diverge. There's certain possibilities for how they might have diverged, and we can systematically explore the space of all those genotypes. Okay? And again, we can't explore all the genotypes, but we can at least explore a large fraction of the different possible combinations of the evolutionary steps that have to happen. And what you can see is kind of what you'd expect, that most of the configurations here have strong negative fitness consequences. So what I'm showing you in this graph, I should explain this more clearly, each node in this graph represents a genotype, and the color of the nodes in this graph represents the fitness measurement that we made using the flow cytometry assay that I showed you before. Okay, and so the red circles indicate cells that have lower fitness than the extant cerevisiae wild type, which we sort of define as the white color. And then green would be some fitness benefit. What you can see is maybe there's a tiny fitness benefit, but overall there seems to be a neutral path through this fitness landscape, or through this genotype network. And even though, of course, most of the configurations are deleterious, there is a path, and again, we don't know which path actually happened in evolution, but at least there is a plausible path here that can take you from the ancestral regulatory network to the extant. Okay, so just to summarize that we've been thinking about gene duplication and protein divergence in the context of a regulatory network, and we found no evidence that these reoccurring evolutionary changes in the protein organization, namely the loss of the kinase in one copy and the loss of those motifs in the other copy, are beneficial. Okay, so what we see is basically all consistent with just relaxation and constraint in some kind of neutral evolution. Okay, so if we take this as a case study of how signaling networks become more complex, what we're saying here is that you just duplicate the nodes in the network, and then you subfunctionalize, you split up the ancestral function, and you build a more and more complicated network that way. But I hope it was clear that we found these things by looking for losses of motifs. And it's certainly possible that we're biased to find this kind of evolution as opposed to finding the evolution of truly novel signaling interactions that also could be underlying the increase in complexity in these pathways. So in the very last few minutes, I will try to tell you a little bit. So I just told you about how an ancient regulatory network, the spindle checkpoint, could be evolving through gene duplication and divergence of short-lived motifs, in this case maybe neutral evolution. And I'm going to try to tell you now about the evolution of novel signaling interactions. Okay? So our hypothesis is that although we can find lots of examples of short linear motifs getting lost over evolution, there should be also cases where new motifs are appearing to make new connections between regulatory networks. Okay? And again, this is just showing you that the pathways are sort of getting more complicated over evolution. I'm going to skip sort of the long story here, except to tell you guys that in collaboration with Eric Weiss at Northwestern University, we identified, using a series of bioinformatics and a lot of biochemical experiments, a docking motif for a kinase in use called CBK1. Okay? And once we, so the other thing so this is a short linear motif that is important for the substrate specificity of this kinase. Okay? And so once we had this docking motif, as well as the consensus phosphorylation site for this kinase, we combine them, and we use this bioinformatics method that a grad student, Andy Lai, developed in my lab a few years ago to predict evolutionary conserved examples of both the docking motif and the kinase consensus site. Okay? And I'm just summarizing that information here. This graph shows you that there's the more conserved docking site you have, the further on this axis you'll go, the more conserved phosphorylation consensus motif, the further on this axis you'll go. And what you can see here is that there's a handful, or maybe 10 proteins that have conserved examples of both of these motifs. And importantly, among those are the 2 known substrates of this kinase. Okay, so very little was known about this kinase before we started working on it, but now we believe we found most, or at least a large fraction of the substrates of this kinase. And the key thing about, okay, so we have the docking motif, and now the key thing is, again, that's just some evidence, is that our collaborators solved the structure of this kinase and actually figured out where the docking site is on this kinase. Okay, and it turns out that the docking site on this kinase actually evolved along the lineage to the fungi. So this kinase exists in human, it exists in other animals, but those species don't seem to have this docking site in their kinase domain. Okay, and amazingly, we could identify some of these substrate proteins in yeast even before the evolution of this docking site. So what this allows us to do is to actually watch, in some sense, the substrates of this kinase go from the ancestral configuration where they contain only the phosphorylation consensus site to the extant configuration where they contain both the docking site and the consensus site. So that's sort of our hypothesis that these new sites are appearing in evolution. And sure enough, we could find examples of these. So here are the here's 3 examples of extant proteins that we believe are substrates and their homologs. I guess these are all orthologs. And what I'm now adding on to these pictures is the evolutionary tree that shows you where we infer the first appearance of these short linear motifs during evolution. Okay? And what I hope is clear from at least these 3 examples is that the phosphorylation consensus site always appears first, and then the docking consensus site always appears second on that evolutionary tree. So the docking consensus is indicated by the yellow stars, and the phosphorylation consensus is indicated by the blue stars. And so what that supports is this idea that the way these signaling networks evolve is that a protein first becomes a substrate of a kinase, and then once the docking motif becomes available, the docking motif can appear to even further enhance the substrate specificity in this case. So we think this is a model for how new motifs can evolve to create increases in signaling specificity. Okay? So just to summarize everything I told you today, we did some systematic tests of our bioinformatic predictions in a model pathway. I told you about evolution, this very surprising evolution of this ancient regulatory network. And then finally, I showed you very quickly a model that we have for how novel signaling interactions can evolve. I told you a lot of things today, so let me acknowledge a lot of people who worked on this. Current members of my lab, Mitch, Bob, Taranay, actually Sun was not a current member anymore, and Ian all worked on this stochasticity in the HOG1 signaling pathway project. Alex and Andy were graduate students who worked on things that are not in the lab anymore. These are other people in the lab below them, and of course I have to acknowledge the collaborators that we worked on this, especially Eric Weiss and Brian Ye at Northwestern University, who solved the structure of this kinase and have been very interested in this docking motif. Okay, so I'll stop there.
Thanks for that amazing talk. Any questions?
Sorry, I went in and out of your talk here, but the beginning part of your talk, you had made mutations in a domain, and then watched that you had a stochastic population after you made that mutation. My question is, how sure are you that the other domain that is your mutated domain is interacting with hasn't mutated itself to give rise to maybe the 2 separate populations? Or
So that there could be some compensatory mutation in our population.
Yeah.
Well, the only, again, we can't.
You would have to sequence it.
We could sequence it. We can't absolutely be sure. But what we think strongly argues that this is not a genetic effect is that we can take a single isolated colony off a plate and grow it and look at the cells under the microscope, and then we see both green cells and cells that are not green. So we think we've ruled out the possibility that there's additional genetic mutations accumulating. Of course, if there's some very unstable genome or something,
right.
But we don't think that's, again, because this is a signal response, right? The cells only turn on this gene expression once we give them a signal. we don't think that they're under some kind of extreme stress until we do the actual experiment. So we think that that's unlikely, but it's definitely a possibility. Yeah.
All your fitness tests, were they done in neutral conditions? All of them?
So we did, the data that I showed you was, again, in the presence of this DNA damaging drug that we know this spindle checkpoint is really important for the cells to survive. We also did the experiments in other conditions, and I still can't see any fitness effects. We've tried a bunch of conditions, but of course we can't be sure. We can't try everything. But at least in the conditions that we've tried, we can't see anything. Yeah?
So in the model, I'm guessing your bimodal output, you require this recovery adaptation at the top.
Does it require the adaptation? We haven't
If you
explored
don't
that.
have that, would it just be a really noisy process towards transcription?
If we don't have the adaptation at the top, will the stochastic response go away? Yes, I think it will, because, yeah, you're right. Eventually, if we don't have the adaptation, eventually everything will turn on.
Everything will slowly turn on, right?
Yeah.
Unless you have like a really strong feedback.
Right, but there is no negative feedback in this model. So that's right. Yeah. So I think what they call this is they call this inherent bistability or something. There's no feedback loop, so you can't, as far as we can tell, there's no deterministic model,
Right.
an analog of this that will give you the bistability.
Because could it, like, just in terms of if there's multiple downstream pathways, like divergent from it, and then you're competing a recovery, like it's not in the model, and like if you don't know what it is, it's hard to do, but if you're just out competing a recovery pathway that happens at a certain point, like will the GFP, will it turn off
No, so the GFP is stable. So once the GFP is there, it sort of sits around.
for how long?
Well, we measure the cell. So after we do the experiment, we fix the cell. So we think in the experiment that we're doing, there's no loss of GFP. So we just watch the GFP accumulate, and that's it. It's stable within the time period of our experiment.
Right. But is there an adaptation process? Sorry.
Yes. Oh yeah.
So
yes.
like if you leave them in salt after a certain amount of time,
Everything goes back. That's right. So this is again, I didn't have time to mention, but this is a very famous signaling pathway, partially because of this. It displays what's known as perfect adaptation. So that when you treat the cells with, you change the concentration of salt, the cells respond, and you can see various cellular changes in response to salt, but then they adapt and go back to baseline, even though the concentration of salt has changed. So there's been a lot of systems biology modeling work to figure out what kind of mechanisms could underlie that kind of perfect adaptation.
Were there any SIMs close to DNA binding domains in your readout in the hog pathway that affected?
We didn't look. Yes, we did look actually in one of the. We didn't find any new ones in the transcription factors in the pathway. But the proteins that we did find in the sort of transcriptional part, one of them is a transcription factor. In fact, that was a known interaction. There was a known interaction motif in one of the transcription factors, and we found it in our assay as well. And then the other protein where we found this very strong bimodal effect, that protein is actually a kinase that's known to regulate one of the transcription factors. So there are sort of these connections with the transcriptional steps.
But that was at the post-translational level,
Yes,
not
that's right.
affecting the DNA binding.
Well, it could. The kinase could be phosphorylating this transcription factor and affecting the DNA binding directly. But so we don't know. I mean, the other interesting thing about this, though, is that we have other motifs in that same kinase that we can mutate and see small effects, but they don't cause these bimodal and stochastic effects. So there is something special, it seems like, about that particular motif that we've mutated. Again, we don't know what it is.
Well, cool. If there are no more questions, please join us in thanking both speakers. And we have some pizza and beverages next door. I think just pop. And please join us for further discussions. Thank you.